This is Katie.
I'm Vinny, and you're listening to Learn Real Good.
It's a podcast about science.
Yeah, get to know science. Learn something good.
Which we like to do. We're big learners.
Yeah, that's the main reason we made this podcast.
That's true. We like learning and we like it to be fun. That's sort of the conception here.
Yeah, I like both those things.
You do?
Yeah.
What else do you like? Is that it? Is that the extent
That's
of the
whole
list? That's the whole list?
That's it. I don't like eating.
Puppies.
Nope, hate them.
Ice cream.
Yuck.
Wow.
Yeah.
That's devastating.
Unless I'm learning about puppies or ice cream.
Okay. There you have it. That's the loophole. That's how we get you to like those things.
Contractually obligated.
So we've done a few of these episodes and I keep forgetting, we should reintroduce ourselves for people who don't know us. So I'm Katie, and I am a biologist by trade. I have a PhD in biology and I teach biology at a cégep here in Montreal. And I do comedy for fun.
Yeah.
What about you, Vinny? What are you all about?
I teach comedy.
What?
As a profession, and I do science for fun.
Now,
yeah,
you used to do science for reals.
Yeah,
for realsies.
Yeah, I did my master's in physics.
There we go. Credentials.
That's what you wanted to hear?
I like to brag about that.
Sure. About my...
I brag about others' accomplishments mostly.
That's pretty good.
Yeah.
You do pretty good too.
Well, okay. Don't want to talk about it. Let's get back to others' accomplishments. Sure. And we like to start the show once we finish goofing with
fun science facts.
Yes. I tried to make that melodic.
It worked.
I regret it. It did not work.
Oh.
But that's okay. Mistakes are gifts.
That's right.
In science and comedy. Look at that.
Yeah. Do you want to go first today?
Oh my gosh. You know what? You knew I was ramping up.
I know. You're excited to share this one.
Trying to avoid it.
So
it's funny because I had it all. I like to have my research in front of me when I bring up these facts. And on this particular website, apparently I had exceeded my free article view on my computer.
No.
And so I can't pull it up on my laptop, but I have it on my phone.
Oh, nice hack. Sorry, paywall. You got hacked.
I know, it's weird. I feel like this particular website I go to way more on my phone, and yet I have more free views. In any case, I do have it. Now, Vinny, before I get to this fact, because the fact is short, so I'm going to ramp up a bit. What do you... Wolves. Okay, that's what we're talking about. Wolves.
Just going right for it.
Yeah, wolves, as you know, sort of historically, they have a bad rap.
Sure.
As much top predators, I feel, often do.
I mean, I'm personally scared of wolves.
Sure, big teeth. I mean, I grew up in Sudbury, Ontario, where the OHL hockey team was the Sudbury Wolves. And the picture, you remember this picture? It's quite grotesque. Look it up, folks. Sudbury Wolves. It was like a cartoony wolf. Fine, but like a lot of like blood coming out of its mouth. So like
they
wolves.
Put blood on the logo?
Yeah.
Wow.
I know.
All right.
We don't mess around in Northern Ontario.
I guess Sudbury just goes for it. Tough. Right to the jugular.
Yeah. Like a wolf might.
Yeah.
But the thing with wolves is they can often be a good thing. Like many top predators, they can control whole food webs and their absence will be felt. A classic example for ecologists is what happened with Yellowstone National Park once they reintroduced wolves. So to simplify the food web, wolves eat elk, which are like big deer.
What's a food web?
Oh my gosh. How sort of who eats who is connected.
Like a food chain.
Yes, but that a food chain assumes that one species solely consumes another species, which wholly consumes
like
another.
In a linear fashion.
Correct. So food web is just closer to reality.
More like a network of foods.
Exactly. Yes.
Okay.
So wolves eat elk, which are like big deer.
Famously.
Yes. And elk eat
wolves. So the circle is complete.
It's a circle of life.
Yeah.
That's what it's about. They eat plants, like young trees and stuff. So in Yellowstone, after all the wolves were killed, because people think wolves are bad, the elk population exploded.
Right.
Because there are other things that will eat elk, but not nearly as good as wolves eat them. Things like coyotes.
They're great at it.
They're so good at it.
Yeah.
And with all those elk eating the vegetation, it had negative impacts on the rivers, had negative impacts on the fish that wanted to live in those rivers, and then all the animals who depend on them. Anyway, the whole food web was, well, not the whole food web, but if you looked at the number of different species that succeeded In the absence versus the presence of wolves, wolves good guys for the food web.
Okay.
So wolves got reintroduced, fixed a bunch of those problems. Now,
Wow. Great science fact.
that's not the fact.
Whoa, that was all set up. I'm already have my mind blown.
So people have legitimate complaints about wolves, right? Wolves in the city. Wolves
You just said how good they were.
Hey! Right, from a park point of view. But if you're
Okay.
living with the wolves, there can be legitimate concerns.
Who's living with wolves?
People who live in the parks.
Oh.
Or in rural communities where
Okay.
wolves have come in and will eat their livestock.
Yeah.
And, you know, they can be bad. But wolves can save human life. That brings me to this fact. Do you have
Whoa.
any guesses, Vinny, how I'm going to indirectly
Yes.
tell you wolves?
I do have a guess.
What is it?
So people every year get into car accidents.
Son of a...
And so wolves come across and they prevent car accidents.
Now, I'm going to die. Is this your science fact?
No.
Okay. I thought we looked at the same thing.
I was just making that up. Is that true?
I am blown away.
Really?
That is it.
Wolves prevent car accidents?
Yes. Okay.
I was talking out my butt there.
I thought that you found the same fact.
No,
and
I
we're
made
a good
up. I
actor
made up
for this
the
whole
fact.
ramp. Okay, so this is a paper that recently came out and looked at 22 years worth of data in Wisconsin prior to wolves being introduced and afterwards, and found that with wolves, deer vehicle collisions went down 25% and was translated to 63 times less cost if you compare cost to loss of livestock and any sort of other negative impacts of wolves on humans. This had a huge benefit.
Oh, because they were fewer elk to hit by
No,
cars.
deer. So we're talking about deer, but
What? Keep your species straight. Are the wolves eating the deer or the elk? Which one
we'll
is
eat
it?
any ungulate. All
Any
right?
indiscriminate ungulate taste.
That's right.
My
So
name of my band, by the way.
wolves, lots of predators will
Right.
use roads because it's a nice clearing to hunt. And so because they are eating the deer that are along the roads, or just them being there and their smell and their presence keeps deer away from roads, it has a negative impact on accidents.
Wow.
Yeah.
Way to go, wolves.
I know, right?
I'm a big fan.
Yeah, it's always a tricky topic. There's a lot of drama around wolves.
There are people who don't like them and want to hunt them.
Yes, and people who think controlling their populations itself is, horrible. Everyone has an opinion on wolves.
Does everyone have an opinion on wolves? Are there no people with opinions?
Every human on
on
Earth.
Earth has an opinion
It's
about
a known
wolves.
fact.
I guess so.
Elk too have opinions on wolves. Not a fan.
Not a fan, yeah. Famously. Okay, well, I have a fact for you. There are bladeless wind turbines.
Oh my gosh. Okay.
So, wind turbines, famously, are known for their big spinning blades. And so, I guess they're not turbines if they don't have their blades, because the turbine implies the spinning. So, what these are they're like giant rods that stick up out of the ground, and they're more like tubes, I guess. They're hollow plastic or carbon tubes, and they wiggle around in the wind, and they hit a resonant frequency. And inside there's like mechanisms that can adjust the distribution of weight so it matches whatever wind is blowing. And then as the wind is causing these oscillations, there's a mechanical system inside, a series of magnets and wires that's generating electricity. Now, it is not as efficient as a turbine blade wind generator, but these are much smaller.
How small?
Like the size of a room, like 10
Okay.
feet, or 2 meters, I guess, if you're a metric person, as I probably should be. We Canadians are weird about where everything's in meters except for like human scale heights.
Oof, yeah.
It's very strange. So they're about 2 meters tall. They're like basically 2 meter tall tubes that wiggle. They oscillate and wiggle, and they make far less noise than the big blade turbines. They don't require anywhere near as much the same footprint on the ground, because you don't need to anchor this giant tower on anything, and it just sticks on the ground. Cheaper to produce. They are less mechanically prone to breakage, because the giant moving parts on a wind turbine, it takes up a lot of oil, it takes up a lot of energy to make these things, so you can mass produce. So the loss of efficiency on this is made up for by its quantity and its ease of reproducing.
Now, what our audience doesn't know is that Vinny now sells these.
So if you get in now, you can come in on the ground floor for just $1,000, and I'll send you 2 of them. No.
What?
It's a pretty fascinating adaptation because yes, maybe the wind turbines are more efficient if you're like out at sea or in remote areas, but having smaller, scalable, less disruptive wind turbines means you can put them in more places.
Very cool. Is there any sort of relationship between how low an amount of wind they can harness energy from?
There is a cutoff, but generally wind is blowing. Like, it's a pretty free source of...
No, yeah. So sorry, what I meant was I feel like that motion sounds like it's easier to generate energy from lower amounts of wind than the amount that's required to spin a thing.
Yes, but I mean, when it's low like that, you're not going to be generating a lot of... Like, you need a strong wind to generate a lot of oscillation, to generate like a usable amount of electricity.
That is neat.
But yeah, it scales up. So instead of one giant turbine, maybe you have dozens of these smaller ones.
Yeah, people, I find wind turbines beautiful.
Sure.
I mean, I'm concerned about its effects on wildlife because it's been noted that they obviously are not great news for birds, and it's suggested the sound they make can have negative impacts on other things. So I think that research is still ongoing. So that's of a concern, but I don't think they're, to a human, visually ugly, but lots of people hate them.
People hate them. People have fears about them and worries about them. And so any smaller scale adaptation of these technology, I feel like it's a good thing. More tools is always better. I
agree, except in the kitchen. Minimalist with tools in the kitchen.
That's right. You have one spoon and just a pot, and that's it. It's fascinating what you cook.
We're not gadget people. We don't have a garlic press. Anyway, send us your garlic presses. Okay, should we get...
For $1,000, we will send you 2 garlic presses.
With your first 6 wind non-turbines.
Yeah, that's right. Okay.
Well, that's enough goofing around. Let's get to the meat and potatoes of the show.
Yeah.
We have a fantastic guest.
Yeah, no one's here to listen to us.
Well, I hope they don't hate us. They're just going to skip the first 10.
That's all we ask. Just don't hate us.
Yeah. We've got low expectations.
Yeah, but ourselves. Our guest, I'm excited to talk about.
Oh my gosh. Well, I get to talk about someone else's accomplishments, which I love.
That's your favorite thing?
Yes, my favorite thing. So Sujani Gomes is our guest. She is a second-year master's student at the University of Alberta in microbiology and biotechnology. Her area of research is environmental microbiology and microbial physiology with a focus on nitrification and related processes. She was born in Sri Lanka and immigrated to Canada when she was 13, and her career goal at the moment is to finish her master's and become a post-secondary science instructor. Please join me in welcoming Sujani Gomes.
Hey, welcome, Sujani. Thanks for being here.
Hello.
Yes, thanks for being here, Sujani.
Well, I mean, first let's talk about microbiology. I'm assuming from the name it's the biology of things that are small.
Yes, it is.
Got it. All right, that's the end of our interview. Thank you for being here.
No mammoths. Correct?
No mammoths. Although mammoth microbiology would be very interesting.
Oh, imagine that specialization on an extinct animal's microbiology.
Wow.
Yes.
I guess so.
Whoa. The gut
Lots to
biome
learn there.
Yeah.
Well, maybe we can start there. Like, what is microbiology? What does a microbiologist study?
So microbiology, like you said, is the biology of small things. But even within itself, it's a very vast field because microbes can be a lot of things, and everything has its own microbiome essentially. So human microbiology can be, medical microbiology, things that make people sick, which is kind of what most people think of when you think of little tiny things that are living on your body. But they can also be really good things, the necessary things. Like now there's a lot of research on gut microbiome, for example, and how that has a huge impact on your health. But microbiology can also be a lot of different things, because what I do has nothing to do with the human body or animals at all. It's environmental microbiology, because they affect a lot of the processes that happen in nature. So microbes can be everywhere in everything and involved in a lot of different processes. So it's it's a very big field about very little things.
It's a great tagline.
That's a good catchphrase.
That's funny. We started by talking about wolves being demonized, but bacteria too. Yeah, that's where a lot of students, when we talk about bacteria in class, that's where their mind goes. Like bad, E. coli, bad, without reflecting on there's so many good ones and we need them.
Yes.
All right, Katie, top 5 bacteria.
I want to hear Sujani's top 5. But before we get to that, let's talk. So you talk about, we're going to get to this.
Yes.
This is a specific and important. I know nitrogen in the environment is a particular conundrum, but I think maybe a good place to start on this topic might be to talk a bit about the nitrogen cycle, how sort of nitrogen flows before we get to what your cool guys are up to.
Yeah. So the nitrogen cycle, I think I probably learned about in high school and then forgot all about it until I started my master's. So nitrogen is kind of like the third most important element when it comes to living things, because after carbon and oxygen, all of our biomolecules have nitrogen, specifically our proteins, because all of the amino acids have nitrogen, and some of our nucleotides have nitrogen. And as we know, our atmosphere is majority nitrogen gas. But because nitrogen gas is made of 2 atoms with a triple bond, it's a very stable molecule, and it's not usable by a lot of the life forms. So there are specific kinds of bacteria and archaea that fix nitrogen, which they take nitrogen gas and then turn it into ammonia. So that's kind of the first step in the nitrogen cycle. And then there's the process of nitrification, which is where my research falls into, where another set of microbes take the ammonia and turn it into nitrite and then to nitrate. And those forms are, especially ammonia and nitrate, are usable by other organisms, especially plants. They will take up that nitrogen and then turn it into other things that we eat. And then that's how we get our nitrogen, or from other animals. Then there's the process of denitrification, which is when the nitrate goes back to nitrous oxide, nitric oxide, other oxides of nitrogen, and then back to nitrogen gas. So it's a very interesting cycle because it's primarily run by microbes. And there's a couple of other complicated processes that are anaerobic and reductions and such. But in a very simplistic sense, there's the fixation, nitrification, and denitrification. And almost all of this is done by different sets of microbes. So that's kind of how the nitrogen cycle is. And something that I didn't actually know about before getting into this research is that the nitrogen cycle is actually really imbalanced in our world right now.
Oh, no.
Have you heard of the Haber-Bosch process?
Whoa. No. What is Haber-Bosch up to? What are those scoundrels up to?
So that's the process of artificial nitrogen fixation. So basically, because nitrogen gas can't be used by plants, it's a rate limiting factor for plant growth. So you know that our fertilizer usually includes like ammonia and nitrates to give plants nitrogen. And in the early 1900s, 2 scientists, Haber and Bosch, they developed this process to turn nitrogen gas into ammonia by like pressurizing it and such. I don't know the chemical reaction. I just know that it's the Haber-Bosch process and it fixes nitrogen without the microbe.
Okay.
And that has definitely increased our food supply and such. But because it's not part of the natural process, it's kind of been overused, and it has led to a great imbalance in the nitrogen cycle, which is a concern.
Interesting.
Wow. So hang on. Let me get this right.
Vinny's got questions.
We have nitrogen in the air. You can't use it.
Yes.
The stuff in the ground that takes the nitrogen gas, puts it in the ground. The plants go into the ground, eat it, and then they use it. And then we eat the plants, or animals eat the plants, and then we eat them, and then we get it, and then it goes back into the
when we die.
When we decompose, I guess it goes back, and then it goes back into the air. So that's the nitrogen cycle. I got that? Okay, great.
Yeah, essentially.
And then the Haber-Bosch part of it is we skip the whole part of going from the air into the ground. We just like take it out of the air, use a machine, and turn it into the stuff that the microbes will turn into the food for the plants.
We turn it into fertilizer.
And then we stuff it in the ground. The plants love it. Okay, great. I think I followed now.
That was I mean, I have to commend you both, Sujani. That was a fantastic explanation of the nitrogen cycle. And Vinny, you really simplified it.
Well, I mean, you got to when you're dealing with me.
Your field.
This is not my field at all.
And having too much nitrogen is also bad, right? In aquatic environments. So that's so if you were dumping, Haber-Bosch got us to dump nitrogen on our crops so we can get more plants, but then a lot of that washes away, and then that causes algal blooms.
Because the plants in the water eat it.
Sure, yeah.
There's a lot of microbial processes going on that are involved there. And I guess I study one of those processes, which is, it's kind of a paradox because it's called nitrifio-denitrification. Right. So there's nitrifying bacteria, which are, or nitrifying microorganisms, I should say, because there's also archaea that does this. And they take ammonia and turn it into nitrite. And then another group turns it into nitrate. And then there's some bacteria that do the whole process. But I study ammonia oxidizing bacteria. So they consume ammonia as their primary energy source. And they turn it into nitrite. But if they're in like low oxygen environments, they can actually reduce the nitrite. They start using the nitrite that they produce, and they turn it into nitrous oxide. And that is problematic because nitrous oxide is a really potent greenhouse gas. You might know it as laughing gas, which is how I knew it before I started studying this stuff. But it is a really potent greenhouse gas. And so I'm studying how these little bacteria produce this gas.
And so we're putting nitrogen into the ground, and then there's a greenhouse gas coming out of it. That's bad.
Yes, it is.
That's my diagnosis.
Yes. Good job.
Wow. So it's not just CO2 that we're messing around with here.
No. It's methane too. Methane's a much more potent greenhouse gas. The thing with CO2 is there's so much of it.
Right. It's sheer quantity.
Yeah.
Right. But yeah, we don't need this. We don't need this.
No? No, we don't.
So looking at this process, Sujani, is your focus more on like why they're doing it, how much are they making, how to stop these guys from doing this? Sort of what's sort of your work focused on?
My work is focused on understanding the enzymology behind the process, like what sort of enzymes are they using to turn nitrite into nitrous oxide. And my project specifically, I work with a specific strain. And this strain is interesting because its genome doesn't have some of the enzymes that were thought to be essential for the process. So, but it's still able to produce nitrous oxide.
There's a mystery.
Which is saying that either what we understand is, there's some gap in our understanding, like what we think is happening is not what is actually happening, or there's other ways to do the same process. So I'm studying this strain, I'm studying its physiology, and I'm starting to work on its proteome to sort of figure out what sort of proteins are active when it's producing nitrous oxide.
Wow, very cool. So would the goal then be to inhibit one of the enzymes to get them to not do it?
First, we got to figure it out. Well, you skipped the whole step.
I know, but
we have a whole question of like, how is this happening? There's a way of doing it that we understand.
Yeah.
And this a bacteria, it's a bacteria,
yes,
is doing it without that mechanism. Well, we got to figure that out first.
So what would you be doing in the lab recently, Sujani, to address this question? Like, what
if you can get into a lab? I guess.
Oh yeah.
I can get into a lab with very limited access, so limited hours, limited personnel. But what I've been working on first is just confirming that this bacteria does have the ability to produce nitrous oxide, that it wasn't just a fluke. So I've been studying its growth curve. I grow them in these gas type bottles with a very specific media that has ammonia and minerals in it. And they eat the ammonia, start producing some nitrite. And because the bottle is gas tight, their oxygen supply is limited. So as they grow, they use up the oxygen in the bottle, and it hits a stage where there's not enough oxygen for them to keep oxidizing ammonia. And they start reducing nitrite and producing nitrous oxide.
Right.
So I measure at what point do they produce that. I've also been doing this assay where I take grown cells and I give them nothing else but nitrite, and I put them in a low oxygen environment and see if they can indeed produce nitrous oxide. So that's what I've been working on for a little while now.
Very cool.
And they're doing it, so.
They're doing it. First step.
Just confirm that it wasn't a fluke.
Well, that's how science works, right? You gotta go step by step. So many false positives can come out of I remember a big physics result.
Oh, right.
They're like, oh, we detected this thing that was impossible, and it turned out all they were detecting was the microwave being used at lunchtime. Is the signals from the microwave oven were being picked up by their instrument. It's like you gotta do your checks.
That reminds me of my first false positive as a youngster.
Yeah.
I used to wear those track pants that have the snaps down the sides, those Nike track pants. This is going somewhere.
Yeah.
And I noticed when I was walking, I heard a creak, and I was convinced there was something wrong with my knee. And I wore these pants so much that I really thought there was an issue. So I went to the doctor and found out that it was just one of the snaps was creaking. Is that the same thing that we're talking about?
Yeah, that's very similar. Yeah. You gotta make sure the source of what's happening.
Yeah, it took a doctor to crack that code.
Amazing.
So Sujani, how can we know how much nitrous oxide in the atmosphere can be attributed to bacteria? Is there any way to sort of know how much of that is from them? Is it that they're the only ones producing it, or is it just we know they make it, we know it's increasing, perhaps they're contributing to it, or is there any way to measure that you know of?
They're not the only ones that make it. There's also denitrifying bacteria. That's kind of certain bacteria that is their job. But there's also industry and all the regular things when you think of pollution that produce nitrous oxide as well. So I believe there are studies that are trying to measure at least how much of this is from this, how much of this is from that kind of thing. And there are some studies that give us an idea about that. But I think it's a difficult thing to measure accurately because in nature, bacteria exist in communities of all different kinds. So it's hard to isolate one group of bacteria and say, okay, this is coming from this process and this is coming from that process.
And the biotech part of the program you're in, is there any, that suggests to me that when I think biotech, I think manipulation of the genome. Is there anything that you're working on with this project, or is that just part of the program you're in that you're not directly working with?
I would say it's more part of the program that I'm in rather than part of my project specifically. I think the biotech part, especially other people in my lab actually work with methane eating bacteria, which is also cool. And they're working more on how to make methane into useful products. So they do a little bit more biotechnology. My project specifically is a little bit more basic research, trying to understand what's going on.
And so what are some applications of this? If you figure out what's happening, then what? So
nitrification inhibitors are a thing that is being looked at, being developed specifically to prevent this process of nitrifiers producing nitrous oxide. And also because sometimes they will use up the ammonia that were meant to be for plants. And they're also important in wastewater treatment because there's a lot of ammonia and urea in there that we want them to use up. So there are applications, especially in agriculture and in wastewater treatment, to sort of make it into a better situation of maybe not producing as much greenhouse gas. But like Vinny said earlier, I'm still at the got to figure out what's going on step before. And I think of it more not only do we have to figure out what's going on, we have to make sure that what we think is going on is actually what is going on, which is kind of where this strain that I'm working on being different from the other strains, but seem to be doing the same thing. It's okay, we don't want to make decisions based on incorrect information.
Sure.
We want to make decisions based on correct information.
That's how I prefer to do it.
Yeah.
What's cool with this project too is it seems like you're in sort of a win-win scenario, right? Like something's different with these bacteria. Either they're doing a different pathway or they're using different enzymes. Like either way, it's good news for you. Like that's important.
Yes.
That's important news, right? That's a good project to do.
Yes.
And so this special strain, was it like discovered? Like, did someone be like, whoa, what's going on here? Is it from like one crater at the bottom of a mountain in Venezuela? Like, what's so special about this strain? Like, where did it come from?
It was isolated from Greece, actually, and it was isolated a really long time ago. It's called Nitrosomonas communis. But I think the issue is more that we don't really know too much about the different species of ammonia oxidizing bacteria. Most studies have focused on one specific species called Nitrosomonas europaea, and that's kind of the model organism for this group of bacteria. But that one is, I think that was just the one that happened to grow the best in the lab and was easy to cultivate, and it's not a very accurate representation of all the different strains that are out there. There are strains that are adapted to different environments, like, for example, the same strains that are more dominant in an agricultural setting wouldn't necessarily be the same strains that are dominant in a wastewater treatment plant. So I think there was an interesting understanding, okay, how do different strains do the same thing? Are they able to do the same thing? Do they all produce nitrous oxide? And so a student previously in my lab had been looking at some different strains and looking at whether or not they produce nitrous oxide and also like sequencing their genome. And she found out that this bacteria does produce nitrous oxide, but it's missing some of the genes that were thought to be important.
So how does I've never worked with bacteria really, just in like isolated labs and courses. How does one go about, if you're working on bacteria, you just go to a lab store online and you order some? Is that what happens? How do you get bacteria?
Yeah, Amazon. Free shipping if you order this many bacteria.
I mean, I did that with newts. That was my experience. I ordered newts from
What?
Yeah. No, there are actually like Amazon like things where you can through a university or a research institute.
I mean, those have to be like vetted and ethically.
Of course.
Right. Well, not of course. I mean, I would hope so.
Oh yeah, the ethics are intense with any living thing. Yeah.
Are there any ethics boards you have to go through for bacteria?
Not that I know of, especially for the environmental ones. But I imagine for the medically relevant ones, you definitely.
And do you have to keep like everything super clean? Like, how do you? Bacteria everywhere.
Yes, and actually, things getting contaminated is a real issue. But the lucky part for our lab is we work with very specialized bacteria that require very specialized media, and chances of other bacteria being able to grow in that media is unlikely.
Right.
But we do still get contamination from other bacteria that kind of feed on the dead matter. Sort of is, I don't know. We still do get contamination sometimes. And I'm like, what are you growing on? There's only ammonia in here.
They don't care.
What are you eating?
That's another research project.
Yes. What is the contaminant?
I guess this is like a bigger question. On a scale of, say, 1 to 10, how much would you say we know about bacteria in general?
Ooh.
Like a guess, obviously.
And the bacteria are listening.
Yeah, they're going to be like, oh, really?
I would honestly say like a 2.
A 2?
Yeah.
Yeah.
Wow. Yeah, you agree, I guess, eh?
I obviously would defer to her, but I'm not surprised it's a low number.
There's so much we don't know.
There is so much we don't know.
We don't even know how to categorize them, right? Like when you look at like how we categorize animals, I mean, it's not a perfect system, but there is some rhyme and reason to how we do it. But bacteria, like we don't have phylums. We don't know what we're doing with how they are, how closely related they are.
Yeah, because like even if you have what you think is a pure strain and you cultivate it several times, over time it will accumulate mutations, and then maybe you have a strain that is adapted to the specific conditions you're growing it in. Maybe and then is it fair to compare that to the same species that you
Right.
isolated in 1992?
Right.
It's like, it's a real challenge. And actually, I was talking to my supervisor a couple of months ago, and she was just saying that it's very interesting that there are categories of bacteria that are isolated from way different parts of the world, and they have similar characteristics, because like, how does that happen in terms of evolution? Like, did they
Right.
get separated and just retain those genes, or
Like some convergent condition. That's wild. Bacteria.
Bacteria are super cool.
Yeah.
they are?
Yeah,
And
we're
I
all
guess
on the same
we
team.
we all depend on them so much, and we know so little about them.
Mhm.
Yeah, but with I think the understanding or the beginning to understand the influence of the microbiome on so many surprising human conditions, not just gastrointestinal things, but all sorts of
System, like whole human body
Yeah.
system.
Mhm.
So I think that's moving us more towards seeing them not like necessarily a bad guy and part of us, part of
Right.
our own ecosystems.
They're not wolves.
Hey, wolves aren't wolves.
Oh, they are wolves then.
They're kind of like
Whoa.
wolves, I would say, because they seem to be the bad guy, but
Yeah,
But you need them. It's all
Totally.
But you
part of
need
a
them.
system.
yes,
Mhm.
you're right.
So they are wolves.
So how did we get onto bacteria, Sujani?
Yeah,
When you
tell
were
us.
little Sujani, let us know like what was she into?
She was into a lot of things, really. So it's been a winding road for me to get here. I had a lot of different interests. And even when I graduated high school, like I didn't know what I wanted to do.
Sure.
So my first year of undergrad, I actually took classes from anything that interested me. And I was actually in business school.
Right.
And
Okay.
then I eventually took biology and chemistry. I took like international studies and French and economics and all these things. And biology was just kind of the one that stuck, the one that I found the most interesting, the most challenging. And then I was introduced to microbiology as part of the required courses. And it was kind of like, okay, this is what I want to do. I think I really like the lab techniques. I liked the streaking plates and just the lab things. I really enjoyed doing those. I liked looking at the little bacteria like ring brand stains and being able to see them under a microscope was cool to me. And then I was doing a cooperative education program, which is like where you work for a couple of semesters. And I had to do a presentation on Idionella sequiensis. I'm probably pronouncing that wrong, but it's this bacteria that can degrade plastic. And it was from a paper published in 2016 by a group of Japanese scientists. And that was kind of like like an enlightening moment of like, oh, they can be an answer to things. Microbes degrade everything that we think is decomposable. And there was this microbe that could eat plastic. And it was kind of like, oh, what else can they do?
Yeah.
Mm-hmm.
And that's kind of how I got into this field of like environmental microbiology. Like microbes can do things that we don't expect them to do. And I was interested in the methane projects that were going on in our lab, because they do cool things where they take methane and turn it into bioplastic precursors and stuff.
Whoa.
Bacteria,
3D print with them.
3D print bacteria.
Yeah, kind of. And then I guess my lab is split into the methane side and the nitrogen side, and I ended up in the nitrogen side. Yeah.
Are you sad about that, Sujani? Are you like longing to cross sides into methane?
Or is this like a sharks and jets?
I have grown into loving the nitrogen cycle.
Like a bacteria would.
Yeah. But I guess it's just not as cool to be like, oh, my microbe can fix your problems with greenhouse gas. Instead, I get to say these microbes are producing greenhouse gas and we need to figure out why.
It's still pretty cool.
Yeah,
it's cool.
I like to think of this like hierarchical class system within microbiology.
Yeah. Wait, so what are the coolest bacteria?
Ooh, yeah.
Well, I think the methanotrophs are really cool because they eat methane and they can turn it into a whole bunch of other things. Yeah, my lab mates all work on how do we streamline this process? How do we scale it up? How do we actually make this a doable thing? But I also think the bacteria eating guy is really cool. I haven't followed up on that research. I probably should, because I don't know what happened with it.
Yeah, the plastic eating bacteria sounds fascinating.
Yeah, and there's also different bacteria that can eat a lot of different things that you just think would not
be edible,
be possible.
Katie, do you have a favorite bacteria?
Oh my gosh. Do I have a favorite bacteria?
Do you have any bacteria you have any special relationship with?
I would defer to stromatolites.
Stromatolites.
Right? So that refers to those rocky structures made of alternating layers of biofilms of bacteria and then like silt, and they build these structures, and they're the living thing for which we have evidence of the oldest life forms. So they have been that type of organism has been dated back like 3.5, arguably 3.8 billion years ago.
I'm on the 3.5 side. Thank you very much.
I like your fake intense stance on that.
I like that it's arguable. Hot argument.
It is.
Well, I guess so.
Well, it's the origin of life, right? Or what we think to be the first.
And so those things, they're still around, and they're still making these pillars, basically.
Yeah. They have fossilized ones near Ottawa, which is, I'm dying to see them.
Okay.
Dying.
Yeah, I know. You keep talking about wanting to go see these fossils.
I do bring it up. You know about stromatolites, right, Sujani? Yeah. They're not on her list, though. She's into way cooler bacteria.
Yeah. What do stromatolites
What
eat?
are they doing?
What do they eat, huh?
Yeah, nothing cool.
Yeah. Get out of here.
My favorite bacteria has to be my strain. That's like my child at this point.
Of course, yeah.
And what do they look like? Are they rod shaped? What type? What do your bacteria look like?
Mine actually are spherical. My friend actually did TEM images, so transmission electron microscopy images, and they were spherical, which I didn't know. And that was very interesting. But they're also like pink.
That is cool.
Yeah. I also grow them in like a pink media because it has an indicator. So I thought they were just the color of the media, but they're not. Even if I grow them in clear media and I like spin them down and concentrate them, they're like a little like peachy color, which is very cute.
That's adorable.
Yeah.
And they don't smell bad.
And they don't smell bad. That's good if you have to work with them for years.
Oof, yeah, some are nasty.
Well, I mean, methane can't be great to work with.
Well, I mean, even growing E. coli in a little petri dish, they smell like really bad breath.
Yeah.
Yeah, they can get stinky.
Well, I mean, it's good that we have a response to E. coli that is not positive.
Well, we have E. coli, again,
I guess you're right.
They're like a wolf.
They're the wolf.
Everyone thinks they're bad.
But they're not. You need them.
Like C. diff, right? C. diff's bad if it's like the dominant bacteria, but it's a normal part of your gut bacteria.
E. coli is also like the model organism for like all bacteria. So
yeah, I didn't want to pick that as my favorite. I feel like that's a hack pick, right?
Yeah, it's like picking the Beatles as your favorite band.
Yeah, we get it.
Yeah, you heard a song.
Now, back on topic, Sujani, what do you, when you look ahead, you said you wanted to be an instructor. So are you talking about being a professor or just any type of instructor? How do you see Sujani's future?
I would like to be a professor. I don't think I want to stay in research. Like, I don't want to be a principal investigator and like run a lab, but I really enjoy teaching. So the best part of my graduate experience so far has been the teaching assistantships. I mean, my graduate experience has also been, interrupted and disrupted by
a very difficult time.
by unprecedented circumstances, but the teaching has been something that I really, really enjoyed. So that's kind of where I want to go. I want to be teaching science, basically, hoping they learn real good.
Oh, nice.
I feel that. And did you see that as being something you might be interested in prior to being a TA?
Yes, I did. I was looking forward to the teaching assistantship as well. I had been like a peer tutor previously when I did my undergrad and also in high school. And a lot of my family have been teachers. My mom was a teacher. My grandparents were teachers. I think baby Sujani, even though she had no idea what her interests were, being a teacher was a cool thing to be.
Yeah.
So Yeah, I would love to be teaching science. I would love to be a lab instructor and show people like how to plate things and
Oh,
stuff.
Are you more like a hands-on kind of person? You like doing the stuff?
Um,
50-50?
50-50.
Yeah.
and so has your teaching then been all or mostly over Zoom, or are you teaching in person?
For the past year, it's been over Zoom.
Mm.
They did have like limited in-person experiences, but they were not really comparable to a proper lab.
Oh yeah.
It was my point of view, but perhaps they were for some courses. I don't know. Teaching over Zoom was interesting.
Mhm.
I don't prefer it over teaching in person for sure.
Yeah.
I really miss just like connecting with students and
Mhm.
like being able to remember their faces, like associate names and faces. So teaching over Zoom was not the best experience, but I'm glad that there is a technology that allows
Yeah.
us to do that.
Absolutely.
At all, yeah.
So
Yeah, it's crazy to think. Yeah, what this would have felt like, or what this would have been like, the effects
even
it would
like
have.
10 years ago.
Yeah, absolutely. Yeah, this is all very, very new.
And thankfully the tools were there to just wait to be picked up. I
Oh my
guess.
gosh. Yeah. And
Now-
if you liked teaching, not as much, but you still liked it over Zoom, that bodes well for you as a teacher. If you didn't absolutely hate the Zoom teaching, that probably means
You're on the right track.
yeah, exactly.
Sujani, what do you like to do when you're not talking bacteria? That's the question.
She's always talking bacteria in the lab,
Yeah.
at the bar.
I don't have any like committed hobbies. I find I think I just pick things up that I want to do for fun, and then I try it out. And if I like it, I do it for a while. If I don't like it, I don't.
What have you been picking up lately?
Lately, I've been picking up wedding planning.
Oh, well, that's a big
Yes,
project.
you're newly engaged.
I
Congratulations,
am.
Sujani.
So my time is very much spent researching and looking up all things wedding related, which I really enjoy. I actually really enjoy like event planning. Like I like throwing birthday parties and baby showers and stuff like that. And I love weddings. I've always loved weddings.
Oh,
So
amazing.
this is my main hobby at the moment.
I mean, also, this is the one where you get the final say.
Yes.
It's not someone else's. This is yours.
Yes.
Will there be bacteria themed gifts at this or...
I hadn't thought about that, but maybe there should be.
Yeah, some table centerpiece.
Yeah.
People can take home a little model of your bacteria.
Ooh, you can use the color of the bacteria for the bridesmaid's gown. Just put it out
Yes.
there.
Yes,
A nice
that is a
peach.
great
Peach.
idea. They're
You're
actually
writing this
cute
down.
color. Yeah.
This is her plan.
I'm sorry.
Let her
I got
do it.
excited.
Yeah.
I like to celebrate other people's engagements. It's in line with my preferences.
Celebrating other
Yeah.
people's stuff.
Well, I think we're near the end, David.
Yeah.
Oh, Sujani, you've been an amazing guest. I have no doubt you are going to figure out this enzyme
Yeah.
sitch on your bacteria. And
It's a great mystery to solve. I'm excited.
I know I'm gonna write this down. I want to follow up and see
Yeah.
what the truth is, because we teach different like alternative metabolic strategies and stuff.
Mhm.
I'll give a shout out to your little peachy boys, whatever these bacteria are called.
Peachy boys. I
Peachy
like that.
Peachy boys.
Yeah.
I'm gonna call them that from now on.
Nice. We did
Peachy
something
boys.
useful.
That's right.
Well, Sujani, you've been an amazing guest. You'll be an amazing teacher, and best of luck with the rest of your masters.
Yeah.
Thank you. Thank you so much for having me. This was such a fun time.
Oh,
Oh,
good.
yeah. We had a blast. Thanks.
Yes. Oh man, that was so
That
cool.
was fascinating.
It's so cool to think of like how like all the different things or living things have realized they can use for food.
I mean, if there's something that can be used by food, something will come along and make it into food.
That's basically it.
That's basically it. That's
Like-
got to be the way the world works.
And like how that works. So this is like the one topic that I teach that I just love teaching because it's just such a rad story. The idea of like respiration, how you take like a sugar, you take glucose, which has a bunch of hydrogens, you rip those hydrogens off, give it to other things, and that eventually powers making energy that your body can use. It's such a wild story, and organisms have learned to use all sorts of things to to transfer that
It
energy.
feels like it's just what's lying around. How do I get it out of that thing?
Yeah.
And use it myself.
Well, there's a huge selective pressure, right? If there's a pile of food that no one's using,
Yeah.
if someone can figure out how to use that, you've got no competition.
And if it's one person's waste can be turned into another
Hey,
thing's food,
you got yourself a cycle.
You get yourself a cycle right there.
I don't know who those characters
They're
are.
from New York. They love nitrogen cycles.
That was our bad New Yorker into
No,
nitrogen.
those were our good. New
Oh,
Yorker.
that's not good.
That's not a good
That's
sign.
not a good
No.
sign. Oh my gosh. Well, that was a really cool story.
Yeah, super fascinating research. Sujani was a lovely guest.
Yes, you got really jazzed there.
I love that stuff. I learned.
Real?
Good.
No, that's nice. Okay, well, Vinny, why don't you send our audience off with our socials?
If you want to follow us on social media, you can check us out on Facebook, Twitter, and Instagram at LRG Pod. That's LRG Pod on all your social media.
You turn into an NPR
Yeah.
journalist at the end. I was going to say spokesperson.
Yeah, I'm also a spokesperson for NPR.
And wind turbines.
And that's right. I love wind turbines.
We get it.
$1,000. You can
Oh my
own your
gosh.
own piece of a wiggly carbon tube.
I hope they sponsor us. That'd be hilarious if we got the weirdest sponsors.
Wind turbines?
Yeah.
Our audience is the people who are going to buy wind turbines.
Yes.
Not like mattresses or like envelopes. Wind turbines.
I want to get that company I bought newts from to sponsor us. That's my goal. I have a goal.
All right. Well, thanks for listening, everybody.
This is Katie and Vinny, hoping you learned real good. Bye.